Okay, thanks very much. My name is Josh Baumgardt. I'm a professor of computer science here at the University of Vermont. And I'm sorry that we couldn't all be together in person. And I'd also like to apologize for the fact that I'm going to give this presentation in English rather than French. You'll have to take my word for it. Your English is much better than my French, so my apologies. What I'd like to do today is introduce you to a new and emerging technology known as computer-designed organisms, or CDOs. This is a technology being developed by myself and Sam Kriegman here at the University of Vermont, and our two biology colleagues, Doug Blackiston and Mike Levin at Tufts University. I'm going to walk you through this technology, but just to give you an idea of how this technology works, it is a marriage or a connection of AI and biology. Briefly, what we do is adapt an AI method to digitally design virtual organisms. And what you can see on the left here is one of these AI or computer designed virtual organisms. Once the AI has designed a virtual organism, we send that blueprint to our biology colleagues, and they then build according to that blueprint, cell by cell, a physical organism according to the blueprint, and thereby they realize, or we realize, a computer designed organism that is different from every other organism that has existed on the planet so far. All organisms, as we know, were evolved by natural selection. The organism that you see on the right was designed by a computer. That's what I'm going to talk about today. So we published our first paper describing computer-designed organisms a little over a year ago, back in January 2020. When we published this paper, there was considerable media interest. We had appearances on CNN. The work was covered by the New York Times, by the BBC, and in actually this month's issue of New Yorker, there's a deeper dive into this technology. However, a couple of months after our technology was introduced to the public, there was another small creature that came along and captured the public's interest, and that, of course, was the COVID virus back in March 2020. However, despite that, over the past year and a half, there's been growing interest from members of the public, philanthropic organizations, funding agencies, biotech companies, AI companies, startup companies in computer designed organisms. So I'm here today on behalf of our team to introduce you to this technology for your consideration. As I mentioned before, computer designed organisms combine AI and biology. So in the first part of this presentation, I'm going to walk you through the AI and the digital design side of things. And in the last half of the presentation, I'll introduce you to the biology side of computer designed organisms. So let's start with AI. As we all know, we're in the midst of an AI revolution. What is driving that revolution? There are many things that are driving it, but at the heart of the AI revolution is a particular AI technology known as a neural network. You might have heard of deep learning or deep learners or the deep network revolution. I'm going to talk briefly about what neural networks are and how we adapted them to create computer-designed organisms for us. There are lots of different kinds of neural networks, but basically they're made up of three parts: the eye, the brain, and the mouth. Let's start with the eye. In a neural network, the eye is exposed to some image that we present to this layer of the neural network. In this case, we can all see that this is a cat, but from the point of view of the neural network, all it sees is a bunch of colorized pixels. Those colors flow from the eye through the inside of the neural network, which we can think of today as the brain of the neural network. And those colors are transformed as data to this final layer, which we can think of as the mouth. In this final layer, if this neural network lights up one of these neurons, one of these parts of the neural network, that is the neural network signaling to us that it guesses that what it is seeing is a dog. In this cartoon example here, we're giving the neural network just two choices. It can guess that it's looking at a cat or a dog. As we can see, in this case, we know that the neural network has made a mistake. So we are now going to teach or train the neural network to not make that mistake again. The way we do that is to start at this incorrect neuron and work backwards. We're going to flow information, or we're going to flow this mistake back from this part of the network, from the mouth to the brain and to the eye. And along the way, we are going to detect certain errors that the neural network made and alter the neural network a little bit. We're going to make slight improvements to the neural network so that the next time we present the same image to the neural network and we flow those colors through the network, in this case, that information triggers the lighting up of the cat neuron rather than the dog neurons. We've corrected the neural network's mistake. If we present millions of images of cats and dogs and humans, and we repeat this back and forth process of teaching the neural network, we eventually end up with a neural network that can correctly recognize cats and dogs and humans and all the other things that the neural network technologies in our computers and smartphones can now recognize. That's AI. In just the past few years, however, we have figured out how to invert or reverse this process to create creative AI. So instead of analyzing patterns, we can reverse this process, and instead of presenting an image to the eye of the neural network, we can instead take one of these output neurons, one of these mouth neurons, and stimulate it. We can press on it like the key of a piano. And by pressing on that key or stimulating that neuron, we flow the information backwards from the mouth to the brain to the eye. And we can then read out what pattern arrives at the eye layer of the neural network. And those patterns tend to look something like what you see here. This is what's known as dreaming networks or deep dreaming or deep fakes. You've probably heard of these technologies. This is the basic idea of how it works. Once you've trained a network to recognize cats and dogs, you can ask the network what a cat is by stimulating this neuron and reading out from the eye of the network. If instead of stimulating the cat neuron, we stimulate the dog neuron instead, you can see this network dreaming about or hallucinating about dogs. If we treat this part of the network as a piano with a whole bunch of keys, and if we press on those keys in some sequence, we can generate a series of images or a series of frames. We can then stitch those frames together into a video. And you might have seen some of these videos online. You get a neural network that in this case is dreaming about cats and dogs. So what we've done is moved from AI to creative AI now, an AI that can generate or design, in this case, images or movies. I'm going to show you how we now take this creative AI and teach it how, instead of generating images or movies, to generate virtual organisms. The first step in this process is to modify the eye layer of the neural network. Previously, it was just a two-dimensional image. We now change it into a three-dimensional image where instead of pixels, we now have what are known as voxels or volumetric pixels or 3D pixels. We're going to represent these voxels as very small cubes. And if we stack all of these voxels together, we have a 3D image. If we stimulate one of the mouth neurons and flow that information into this cube, the neural network will generate or paint colors onto these voxels, onto these 3D pixels. And in the same way we just did with cats and dogs, we can stimulate different neurons in the neural network. And every time we do, it will paint a different pattern onto our 3D image. So now we have a neural network that can generate 3D images. We're now going to make the next alteration, next change, which is to treat this three-dimensional structure not as an image or an animation or a video. We're going to treat it as a physical structure. We're going to assume that each of these small cubes is a physical cube. It has a size, a mass, a weight, and so on. The color is going to tell us what we should do with that physical voxel. In this example here, we're going to assume that all of the voxels that are painted gray, and you can see most of them are painted gray, we're going to remove those gray voxels. So you can see here in my little cartoon example, we're starting to remove the gray voxels. If we remove all of them, we're left with a 3D structure. So now we have a neural network that by pressing on the keys of the piano or changing the stimulation or stimulating this neural network in different ways, it can generate different three-dimensional shapes. At the moment, these are just 3D shapes. We need to now take another step where we turn these 3D shapes into virtual organisms, things that can move. So in order to do that, we take these 3D shapes and we place them inside of a physics engine or a physical simulator. If you've ever played a modern video game where you're running around inside a virtual environment and objects have mass and they fall over, things blow up and fall back to the ground. If you have a virtual world with physics inside, that's what you're looking at here. So we've taken our 3D structure and we've placed it inside of this virtual physics engine. And you can see that we've now gone from a 3D shape to a 3D creature, something that is able to move. Okay, so now that it can move, we can take another step forward and we can ask the AI to design not just an arbitrary 3D moving object, but we can ask it to design a 3D object that moves or does something useful. The way that we do that is to tell the AI to create something like you see in the bottom left here. All of the question marks mean that we don't know whether a voxel should be placed at that position or not. We don't know what the shape of this creature should look like. We want the AI to design the shape of a creature so that when we put it inside the physics engine, it will move as quickly as possible from left to right. So we're now asking the AI not just to design a structure, but to design something that does something useful for us. It turns out that there is an almost infinite number of shapes that the AI could create. So among the space of all possible shapes or all possible virtual organisms that the AI could create, how does it search through this space to find the best shape that moves as quickly as possible from left to right? That's a search process. In order to do that, we're going to make a small change, which is we're going to change the way that we think about this neural network. A neural network is basically a piece of code. We call it a neural network because it kind of reminds us of brains in the way that it works, but we can think about it in different ways. So I now want you to think about this piece of code that is able to generate virtual creatures. I want you to think about it not as a brain, but as digital DNA. So in the same way that our DNA creates us, I want you to think about this computer code as digital DNA that generates or builds virtual organisms. So here's an example of one piece of digital DNA that produces a virtual creature. Imagine now that the computer creates not just one piece of DNA, but it creates two pieces of DNA. And these two pieces of DNA are different, which is shown by the light and gray, the light and dark gray in these two different pieces of DNA. If we read out these two different pieces of DNA, they create two different virtual creatures. In the same way that your DNA and my DNA have produced two slightly different humans, here we have two separate pieces of digital DNA that produce two different virtual creatures. Remember that we've asked our computer to design a virtual creature for us that moves from left to right as quickly as possible. If you watch these two animations, you'll notice that the bottom robot moves to the right a little bit faster than the one above. So in this competition, the slower moving robot is deleted, and there is only one surviving organism in this case. We now have an empty slot in the population. We take this digital DNA, we make a copy of this digital DNA, and after we've copied this digital DNA, we make a small change in the DNA. We introduce a small mutation. Now that we've changed this digital DNA, if we again stimulate the left side of this structure and ask it to generate something, now this new piece of DNA produces a new virtual creature, where the creature at the top is the child of the parent creature at the bottom. You'll notice that both creatures have sort of similar structure, not quite the same. The child is not quite the same as the parent. And in this case, the child organism is moving a little bit faster from left to right than the parent. So in this case, the parent loses the competition, it's deleted. The child's DNA is copied, mutated. We simulate the grandchild robot, and on and on and on. This algorithm that I've just described to you is known as an evolutionary algorithm. Just to step back for a moment, what does this algorithm do? It generates a population of DNA or pieces of computer code. Each one of these pieces of computer code generates its own virtual organism. The computer simulates each one of these virtual organisms. It deletes the slow-moving organisms and makes randomly modified copies of the faster-moving organisms. That's how we get the computer to digitally design virtual organisms for us. Okay. That's the AI side of things. I'm going to switch now and talk about how we move these digital designs into biology. I'm going to start with a particular organism. This is the African clawed frog, otherwise known as Xenopus laevis. Turns out that we can actually take cells from Xenopus laevis and rearrange them into new structures. And I'm going to describe what I mean by that. To start, we harvest skin cells from not the adult frog. So in this image here, it looks like we're taking skin cells from the adult frog. As you'll see in a few minutes, we actually harvest skin cells from very early frog embryo, basically frog eggs. So no adult frogs were harmed in the making of this research. We're going to take skin cells from frog embryos, and we're going to represent those skin cells as these light blue voxels. So we're going to tell the computer again that it can design these virtual organisms, and wherever it places a light blue voxel in the physics engine, that light blue voxel is going to be treated like virtual skin. Skin can be stretched and pulled, but it is not able to cause any actions. It's passive material. We are also going to harvest another cell type, again from early frog embryo, not from the adult frog. This cell type, these cells are heart muscle tissue, or heart muscle cells. We're going to allow the computer to simulate heart muscle cells as basically voxels that alternate between red and green. These are voxels that if they're placed inside the virtual organism, these little voxels are going to increase and decrease in volume, very much like cells do in the frog heart and the human heart. If you put frog heart cells together in the shape of a frog heart, or you put human heart cells together in the shape of a human heart, all of those cells will increase and decrease in volume and pump blood in and out of the heart. So basically what we're telling the AI is it can make virtual organisms, and it can make it out of two different types of Lego bricks, Lego pieces. The blue pieces and the red-green pieces that increase and decrease in volume. The evolutionary algorithm goes to work, and what you're seeing on the left now are 3 pieces of digital DNA that have been translated into 3 different virtual organisms. You can see that in each of these initial organisms, the combination of blue cells and red-green cells are different. Up till now, I've been referring to these virtual organisms as computer-designed organisms. They have since been nicknamed Xenobots because Xeno is short for Xenopus laevis. So for the rest of this talk, I'll refer to these little creatures as Xenobots. As I mentioned before, the red-green voxels are meant to simulate heart muscle tissue. So you'll notice that the red and green voxels are increasing and decreasing in volume, growing and shrinking. And you'll notice that the blue cells, the skin cells, are passive. They're being pulled and pushed by the heart muscle tissue. We've asked the AI, in this case, to design a Xenobot for us that moves from left to right. You'll notice that these first 3 Xenobots that the AI has dreamed up don't move at all. However, the second and the third one are moving a little bit more than the first one. So at this point, the computer is going to delete the upper left Xenobot and make randomly modified copies of the surviving second and third Xenobot. And if we repeat this evolutionary process over and over again, and we ran this in our supercomputer here at Vermont for a few days and then a few weeks, the AI eventually produced this virtual creature, which, as you'll see, is able to move from left to right. It doesn't move very fast, but it definitely gets the job done. So this was the best design that the computer could come up with, given only these 2 cell types, these 2 different types of building blocks that the computer was able to work with. We then took that design and we gave it to our biology colleagues, and they started to construct this virtual Xenobot in reality. And I'm going to show you just a few short videos that are taken from the construction process. What you're seeing is you're looking through the microscope as our microsurgeon, Doug Blackiston, is going to go about trying to build this blueprint in reality. You'll notice there are a couple of spheres here, or balls. These are, as I mentioned, early frog embryos. So these are basically frog eggs. A female frog will lay millions and millions of these eggs. So we've harvested a couple of her eggs. Since she lays several million eggs, she's probably not going to miss a few of them. You'll notice that Doug is reaching in with micro forceps, and he is gradually cutting off the top of the embryo. Inside that embryo are stem cells, and that's what Doug is going to build this Xenobot from. In this next step, he's cut off the top pieces of 5 of these embryos. You'll notice there's some dark material, and as you watch this video, you'll notice that Doug is slowly removing the dark material away from what lies underneath, which are stem cells. If you look closely, you'll be able to notice that you can actually see each individual cell in this case. The magnification that you're looking at, the total width of the video in this case is about 2 millimeters across. So Doug is a very accomplished microsurgeon. He has very steady hands. This is very, very difficult, detailed design work. So he's separating off the dark material, which is ectoderm. It's a part of the skin of the early embryo that we don't need. And we're going to harvest these pure stem cells that lie underneath. Once he's removed all of these stem cells, he pulls them up into a syringe, and then he places the syringe back into a dish, and he injects these skin cells into a small cup. I'm just going to start this video over for you. You'll notice this dark circle. This is a small depression or a small cup. We're looking down onto the top of the cup. And in this fluid, these stem cells are going to gradually fall into this cup, and then gradually they are going to start to pull themselves together again. Biological cells do not like to be left on their own. So as they sit in this cup for a few hours and a few days, you'll notice that they start to pull together. These stem cells attach to one another, and they pull themselves inward and become increasingly dense. So what Doug is doing is creating the raw mass, the raw material that he's going to use to start sculpting or start creating these Xenobots. You'll notice now that we now have this ball of pure stem cells. Doug is now going to reach back in with forceps on the left and what's known as a cauterization device on the left. The cauterization device is basically just a hot wire, and that hot wire is burning away or removing some of the material that's not needed. As you're watching this video, I'll give you a few more details. As I mentioned, we're looking through a microscope down into a dish. That dish is filled with room temperature fresh water or pond water. Frogs tend to like warm, fresh water. So not surprisingly, frog stem cells are also perfectly happy to hang out in room temperature fresh water. You'll notice that Doug is burning, I'm just going to back up a few seconds here. You'll notice that Doug is burning a small cross pattern into the Xenobot. And what's left over, if I can skip ahead a little bit here, you'll start to be able to see this in the video. These are going to be the small legs that you see that make up the small virtual creature that you see on the left. So he's trying to build as best he can a copy of this creature. Okay, a couple more details I'll just mention as I play parts of this video again. As you can see on the left, the virtual creature has heart muscle tissue on the bottom. These are the red-green voxels. And it has skin cells, the light blue voxels on the top. I mentioned that these are stem cells. There are actually two types of stem cells that are mixed in here. There are stem cells that are going to develop into skin cells. And these are going to be the skin cells on the top of the robot. And Doug also included a second type of stem cells, which are going to develop into the heart muscle cells, and he put those on the bottom. So the little legs that you can see that are pointing up towards us, pointing up towards the camera, those, although they're white, they're going to develop into heart muscle cells, and they're going to increase and decrease in volume. And the creature at the moment is on its back. And its back is going to be made up of skin cells, stem cells that turn into skin cells. Okay. After this process is complete and we leave this Xenobot alone in the dish for a little bit to mature, eventually, after a while, if it's turned upright again, so that now we're looking down through the microscope at the back, we're looking down onto the top or onto the back of the Xenobot, the Xenobot eventually starts moving. And as you'll see, it moves kind of like the virtual organism. They're not quite identical in shape, and they're not quite identical in the way they move, but it's pretty close. I'll play this for you again so you can see this in action. So we've now gone from telling the AI to design a virtual creature that moves from left to right. The AI has come up with an idea. It's come up with a design, which you see in the top part of the video. And if we take that design and build it from actual frog cells, we get a small creature that is able to move along the bottom of the petri dish in the way that the AI predicted, that it would. Just to give you a sense of scale here, what you'll be able to see on the left in a moment is one of the team members, Sam Kriegman. He's holding a small glass plate, and I don't know whether you're actually able to see it at the moment, but if you squint, you should be able to see that there's a small black speck that's captured between two plates of glass. That's the Xenobot that you see zoomed in on the bottom right. Okay, so these Xenobots are about the size of a grain of sand. And as I mentioned, we can get an AI to design these for us. In this very first experiment, obviously walking along the bottom of a petri dish, it's a very, very simple behavior. It's not very useful, but this was very important to demonstrate for us and for others that this technology is possible. A natural question that's probably occurring to you is, so what? What can we do with computer-designed organisms? Well, we've started to explore possible applications. I'm going to show you what happens if instead of putting one Xenobot in the bottom of a dish, what happens if you create multiple copies or you build multiple copies of the same design and let them go in a petri dish. The simulation at top shows us what happens, and it matches pretty close to what happens if we let the Xenobot swarm loose in a petri dish. You'll notice that given the computer-designed shape of the Xenobots, they happen to bump into each other from time to time. I'll just loop this video for you. You'll notice that given their shape, from time to time, they bump into one another and they kind of lock together like a lock and key, and they move together for a while and then they separate. You'll notice that process occurs in the simulated Xenobots at top, and it also occurs in the physical Xenobot in the bottom half of the panel. You'll notice that we did something else, which is we sprinkled small particles of matter in the dish as well. So these small objects are much smaller than the Xenobot themselves. And you'll notice they're a little bit hard to see in the physical Xenobots, but you might be able to see there's some small dots, some small imperfections in the dish, and those are being pushed around by the physical swarm as well. Here's a higher resolution photograph, so you can see sort of the end result of the swarm. In this case, the particles were very small red glass beads. They're very, very small. And you'll notice we distributed millions of these small beads across the bottom of the dish. And when we let a Xenobot swarm loose in this dish, you can actually see the path of some of the Xenobots through the debris. And you'll notice also that these Xenobots push some of this material together into piles. This is kind of one of the interesting things about Xenobots compared to traditional robots, is that Xenobots are made of cells, and an individual biological cell is itself a very complex and intelligent machine. Cells can actually sense their environment, and they can act in limited ways as well. So it may be that although these Xenobots were only designed with skin cells and muscle cells, there's no eyes in these Xenobots, they have no brains, they have no digestive system, they can't eat. They have no reproductive system, they can't reproduce. They're just skin and muscle. But the cells themselves that make up the Xenobots, the cells have eyes and the cells are able to move. So it's possible that these Xenobots are able to, in some way, see their environment and act on that environment in a way that leads to a non-random result, which is the creation of a pile, pushing a bunch of this material together into a pile. So there may be some intelligence that comes for free when we build robots out of biological cells than if we build traditional robots out of dumb materials like metals, plastics, and electronics. So one of the things we're interested in exploring in future is the advantages and disadvantages of building robots out of artificial materials like metals and ceramics and plastics, or building them out of biological materials like cells. This technology is an example of synthetic biology. Xenobots are not the only example of synthetic creatures. Many of our colleagues are getting better and better at applying AI to create machines which are combinations of artificial and biological materials. What's unique in the work that I've shown you today is that each Xenobot is 100% biological. There are no artificial components in these small robots. That's important because it makes this technology biocompatible, biodegradable, and arguably carbon neutral. And so again, that suggests possible applications for this technology in future. I'm going to show you another video now. This is zoomed in on just an individual Xenobot. You'll notice that both the virtual and the physical Xenobot are pushing a single object in their environment. Since these are completely biocompatible and biodegradable robots, as this technology matures, we may be able to create very large numbers of Xenobots, and we may be able to introduce them into environments where there are pollutants. So we could, for example, introduce a Xenobot swarm to contaminated soils. The swarm could find the pollutants and pull it out of the soil. Or again, since these robots are perfectly happy to exist in fresh water, we could introduce, in future, we could introduce this technology to waterways and have these small robots search for microplastics and pull those microplastics out of the water. Those environmental applications are years away. As I mentioned, this technology is just a little over a year old. We're still understanding the advantages and the disadvantages of this technology, but it's kind of fun to think about where this technology might go in future. I'm going to show you another simulated Xenobot here. This one, which again was designed by the AI to move, from left to right. Actually, in this case, the AI came up with a hollow design, because if you want something to move in water, it's a good idea to be hollow so the water can pass through your body. This was kind of a surprise. We weren't really expecting the AI to design a hollow Xenobot. But one of the nice things about a hollow robot is you can place a small object inside it, and then as the robot moves, it transports that object for free. Why is that important? There's another set of applications that we're looking at for this technology, which is not environmental applications, but medical applications. It may become possible in future to design not just Xenobots, but other kinds of biobots built from cells taken from other organisms. It may become possible to build biobots out of human cells rather than frog cells. If we can, we might be able to actually harvest cells from a human patient, construct a biobot out of that human patient's own cells, and then have the patient swallow that small biobot and have that biobot swim or navigate inside the body of the human patient and deliver medication to a desired point in the body. This is merging this technology with a related technology and a related emerging technology, which is intelligent drug delivery. How do you get drugs to specific points inside the human body without performing invasive surgery? If you were to do it using bots made from the human patient's own cells, when you ingest that robot, it would not trigger an immune response. The last video that I'm going to show you is again, an advantage of building robots from cells rather than artificial materials, which is that if you damage the bot or it becomes damaged, in this case, Doug is snipping or cutting this robot almost in half, you'll notice that it heals itself back up again. I'll play this part of the video a couple times. There's the cut, and there it is healing itself again. As we all know, our computers and phones, when they break, they do not fix themselves. But if you build machines from cells, in some cases when they break, they do indeed fix themselves. Okay, I think I will conclude here. I've walked you through the beginnings of this new technology. I'll leave up this URL for you for a moment. You can navigate to cdorgs.github.io. You can find more information there about this technology. And I'm very happy to hear your thoughts on what I've presented today. Thank you very much.